Annenberg Media.
By the National Science Foundation, America's investment in the future. By the American Society for Microbiology. And by the Office of Biological and Environmental Research of the United States Department of Energy. Additional funding was provided by the Arthur Vining Davis Foundations. This program has been made possible in part by the Corporation for Public Broadcasting.
Human beings are teeming with microbes. There are microbes on our skin, in our pores, in our mouths, on our hair, under our nails, not to mention the microbes that squirm around inside our intestines. Really, we're perfect mobile homes for them, each of us carrying our own unique combination of microbes. For the most part, we live in harmony with them. But what happens when the normal balance of microbes gets out of whack?
Disease, that's what Sorry about that. I'm Jim Leinfeld. In this episode of Unseen Life on Earth, we'll explore the relationship between microbes and human disease.
Infectious diseases are as wide-ranging as the microbes that cause them. We come in contact with pathogens every hour of the day. Some pathogens even take up residence in our body, and we don't even know it until we feel sick. Our own microbial flora may become unbalanced. In this case, good bugs go bad, like a yeast infection. Or we encounter known pathogens like salmonella by eating contaminated foods, or by boldly going where no human has gone before. We encounter diseases that are new to us, like Ebola.
Diseases can spread from one person to another if the environment is right for transmission. Sometimes this can be as simple as a handshake.
Some microbes, such as the ones that live in our digestive tract, are beneficial to our well-being. Other microbes, however, have been our enemies for thousands of years. Throughout history, infectious diseases have wiped out huge populations. In 1918, over 20 million people died from an influenza virus that swept the globe. Eventually, we realized that each disease seemed to have a perpetrator, often with distinct attributes such as speed in replicating or agility in beating our immune cell defenses. Then we began to dramatically change the balance in our favor. First came the concept of hygiene, washing with clean water and soap. Public health has improved dramatically thanks to fresh water supplies and sanitation. The discovery of penicillin heralded the age of antibiotics.
Antibiotics, along with vaccines, were the tools of modern science that gave us an apparent edge in the competition against pathogenic microbes.
But this edge may not last forever. Microbial killers like HIV can emerge when something tips the balance in favor of a particular microbe. Other microbes, such as Ebola, the mysterious killer that surfaced in Zaire, or Legionnaire's disease, can appear when something changes in our relationship to the environment. These can be the most lethal. Around the world, few things are feared as much as outbreaks of diseases which kill swiftly and can appear anywhere, anytime. Such was the case in New Mexico. On May 14, 1993, in a rural community of the Navajo Nation near Gallup, New Mexico, an unknown microbial killer swiftly claimed the life of this athlete. His fiancée also died. Within a week, 4 more. Dr. Stuart Levy.
One of my attendings came to tell me that he had admitted a patient that night with a disease that he hadn't seen before.
Dr. Fred Koster.
We had to rule out known viral pneumonias, known bacterial sepsis, septicemic plague.
And suddenly that patient had a cardiac arrest for no explicable reason whatsoever.
The mystery illness caused flu-like symptoms and acute shortness of breath. The victims died rapidly, within 1 or 2 days.
And then seeing this kind of on not a daily basis, but once or twice a week.
Over 50% of the patients admitted died. The lab tests at the University of New Mexico Hospital searching for the cause of the disease were unsuccessful. The disease spread throughout the Southwest. A mother of 2 in Nevada, a man repairing his house in Arizona, a teenager in Colorado. The outbreak was serious enough to start a national alert and bring in the Centers for Disease Control.
We got the call from the folks in the Southwest, and they had reached a point where they knew they needed help. We didn't know what this was. It was totally, absolutely unknown. We knew people were dying. We knew we had a problem with some kind of disease that looked like an infectious disease. It had a very high mortality rate. We didn't have a clue what the actual organism was.
C.J. Peters is the director of the Emerging Pathogens Department at the Centers for Disease Control and Prevention in Atlanta.
We had 15 or 20 suspected patients, and it could have gone to 30, 40, 300. I mean, you just don't know when the curve starts going up until you can get a handle on things. We look for absolutely everything. We look for all the bacterial agents, the viral agents, mycoplasma, fungi, you name it.
Within 19 days, the CDC identified this microbe, a hantavirus, as the dangerous killer.
There are thousands and thousands of viruses. But where did this particular virus come from? How did a seemingly unknown, deadly disease spring up in the U.S. out of nowhere? Well, scientists learned from the Navajo people that this disease had plagued them twice before.
Dr. Ben Muneda, the grandson of a Navajo medicine man, was part of the CDC investigation. He uncovered an early clue.
I went around the reservation to the traditional healers that I knew, and then I asked them about the knowledge of the mystery illness, and what they told me was quite astounding. I was listening to them describing the things almost word per word, we were piecing together in our meetings with the CDC.
The medicine men described the deer mouse as a carrier of the mysterious illness. The deer mouse and the hantavirus have been evolving together over millions of years and have come to an accommodation in which both survive. It is only recently that humans have become part of their story.
The traditional healers told me that the way that the deer mouse will take a human life is that through their urine and their droppings, And the way it passes is through your eyes, or through your mouth, or through your nose.
When hantavirus is inhaled, it will infect cells, allowing it to reproduce. Inside the lungs, small air sacs are surrounded by blood vessels. Within the blood vessels, our white cells patrol, looking out for invaders. Hantavirus, seen here as black dots, infects the cells that line the blood vessels. The white cells sense the invader, push through, and emit cytokines that inactivate the virus, but also weaken the blood vessels. Plasma leaks into the air sacs. Soon, the plasma accumulates, leaving no room for oxygen.
4 years after the first case had been diagnosed in New Mexico, 180 people had died throughout the U.S. and Canada. Over 100 more had been infected.
There are many examples of transmission of a virus or microbe from animals to humans. Plague also comes from rodents. Anthrax from cattle. In each case, something allows the microbe to expand into a new host. Something in the ecosystem tips the balance.
Once it became clear that deer mice were the reservoir species for this new hantavirus, we hypothesized that what caused the '93 hantavirus outbreak, at least in the southwestern United States, was the El Nino-driven increase in plant primary productivity, which caused more food for mice, which caused more mice, which caused more people to come in contact with infected mice, and thus more hantavirus pulmonary syndrome.
We all fear the day when another new microbe emerges, becomes mobile, and potentially sweeps the globe with disease. But what if a new killer wasn't an emerging disease, but a re-emerging one?
"400 years ago, in the English countryside, a mysterious respiratory disease terrorized Tudor England. It was called the English sweating sickness for the high fevers it induced. It ravaged the countryside between 1485 and 1551. The disease even changed the course of history by taking the life of young Prince Arthur Tudor. His younger brother, Henry VIII, became king, paving the way for the English Reformation. Then, the sweat disappeared entirely. For over 400 years in metropolitan London, there has not been a single documented case of the sweating sickness. It was almost forgotten. That is, until 3 medical detectives suspected a connection between the sweating sickness and hantavirus. Dr. Vanya Gant and Guy Thwaites teamed up with Mark Tavener, a 16th century historian who could translate the Old English and Latin of ancient records. The 3 of them set out to solve one of England's oldest medical mysteries. The sweating sickness so affected Tudor England that it was the subject of one of the first books written about disease. It described all the symptoms besides high fever and sweating.
The clinical descriptions, as best described, were of a very rapid onset. One would feel flu-like. You would have joint pains, headache, feel a little bit under the weather, and then a gathering shortness of breath to the point where some 24 to 48 hours later, you were dead.
I said, this sounds exactly like the Four Corners disease, the hantavirus pulmonary syndrome that had been described maybe 2-3 years beforehand in the southwestern corner of the United States.
Thwaites and Tavener set out to find a broader source of information. Their richest came from gravediggers' records and old parish registers.
Parish records of over 680 churches showed the vast effects of the frightening disease. Gravediggers' records revealed 5 outbreaks during the Middle Ages, with most victims succumbing during the hot months of July and August.
It was certainly a summer disease. It was not a disease that was associated with poor conditions of sanitation. It was scattered, it was rural, it didn't conform to any patterns of people-to-people transmission.
The accumulating evidence led Guy Thwaites to be more and more convinced that the sweating sickness was caused by some kind of hantavirus.
What we see in modern medicine is just a snapshot. We don't see anything other than that. We don't look at history in modern medicine. We don't get taught it in our medical schools. Perhaps we should be. Perhaps we ought to get more perspective on the evolution of infectious diseases. And we think that we are seeing them for the first time. But we're not seeing them for the first time. This is a cyclical thing. They reappear and appear, and they've done so throughout the whole of history.
The relationship between the English sweating sickness and hantavirus is only speculative, but over time, pathogens do evolve, sometimes developing new characteristics that make them appear to be different diseases. 8,000 miles away from London, and just as far from the southwestern United States, is Pergamino, Argentina, the breadbasket of South America. But this remote and peaceful community is linked to those parts of the globe by an unknown danger that is lurking in the summer grass. For the local residents, it was a shock to discover hanta appearing in their surroundings without warning in a deadly new form. Dr. Delia Enria is a physician and microbiologist expert on viral diseases. At the National Institute for Human Viral Illness in Pergamino, she noticed unusual symptoms in some patients. She didn't know it was a hantavirus. Through the internet, she received a CDC report about the virus.
You are a detective. You are following step by step what we call an hypothesis.
In September 1996, 3 years after the Four Corners outbreak, Dr. Enria feared that the disease had reemerged in a different form. In the quiet resort town of Bariloche, southern Argentina, 4 people became ill with flu-like symptoms. They were short of breath, and their lungs filled up with fluid. Tests sent to the CDC proved that the killer was another new strain of hantavirus. Over 50% of those initially stricken died rapidly. The outbreak caused panic. Something was happening that had not happened in the United States. People who came in contact with victims also came down with the disease. A reporter covering the story suddenly got sick and died. A doctor who had treated patients in Bariloche became sick. He traveled 1,000 miles to Buenos Aires to see a specialist. The specialist got sick. The specialist's wife got sick. All 3 died. Delia Enria suspected person-to-person transmission, something that had never occurred with any hantavirus outbreak. This would mean the virus had changed, becoming capable of jumping from human to human. If this was the case, the virus would spread easier and faster. At the hospital in Pergamino, Dr. Enria had another case.
I got a call from Dickie Enria at the Institute for Virology in Pergamino, and she wanted to talk about the issues around human-to-human transmission. Well, I figured that that was probably just a fantasy.
Facing skepticism about her person-to-person transmission hypothesis, Delia Enria was determined to find the source of the virus. Even if it had evolved to be contagious from one person to another, the starting point, the original source, still had to be a rodent.
The Argentines organized a team. A couple of us went down to work with them. We began to set up a series of hypotheses about the way these people were getting infected. And we had a long list, and we went through the list, and we were able just to draw a line through each hypothesis until we were left with person-to-person transmission. Not supposed to happen with hantaviruses.
Person-to-person transmission of the virus could have meant rapid infection of nearby Buenos Aires or beyond. A neighbor, a relative, anyone could have passed the virus. Avoiding rodents would not prevent the disease.
The virus diseases that have been able to come into humans, cause problems, and be transmissible from human to human are the diseases that we know we have to watch out for.
Yet, in the case of this hantavirus, the disease mysteriously vanished, at least for now, almost as quickly as it had appeared. Ironically, although the virus changed strategy, its mode of transmission, it was still reaching a dead end. Either the virus kills its human host, or the host kills the virus before it can be transmitted. Humans are not a good host for hantavirus. Yet.
There are several major things, in my opinion, that are changing our relationship with microbes and with pathogenic organisms of different kinds. Climate change and changing human land use patterns.
War, famine, and changes in the microbes themselves also impact our relationship with pathogens.
Now, what's happened in modern times, as shown by the hantavirus, is that human populations have gotten bigger and bigger and bigger. And we suddenly come in contact with creatures which we ordinarily didn't before.
We still have not encountered many of the microorganisms that are out there in nature. And the more we encroach in the environment and the more different living things that we affect, and indeed the more species that we wipe out, every time this happens, we really affect the microbial balance in the world in ways we can't understand.
We fear what we don't know. But as we come to understand the microbial world, we begin to realize just how interconnected our existences are.
Understanding the relationship between humanity, microbes, and the environment is a scientific challenge for the coming decades. Using this understanding to fight microbial disease is not only a scientific problem, but a social and economic one as well. The dance with microbes has lasted as long as we've existed. While some are dangerous, most are not. Besides giving us disease-fighting drugs, many help us make food, give us clean water, and have generally created the environment that sustains us. We didn't know they existed until we saw them through the microscope. Now, these earliest forms of life reveal our past and point the way toward our future. We are locked in an embrace of cause and effect with the microbial world. We do something, and they change. Then we change. And so it will continue for as long as there is life.
Annenberg Media.
By the National Science Foundation, America's investment in the future. By the American Society for Microbiology. And by the Office of Biological and Environmental Research of the United States Department of Energy. Additional funding was provided by the Arthur Vining Davis Foundations. This program has been made possible in part by the Corporation for Public Broadcasting.
For information about this and other Annenberg Media programs, call 1-800-LEARNER and visit us at www.learner.org.